Hello, good afternoon.
Good afternoon, sir.
Hello, good afternoon.
Good afternoon, sir.
So can you hear me?
Yeah, Sharon, good afternoon.
Good afternoon, sir. My internet is a little slow today, sir, actually.
Okay. Okay, just 1 and 2 minutes. Waiting for others to join. Is my voice is audible?
Yes, sir.
Yes?
Yes, sir.
Okay. Good.
So actually for me it's getting stuck right now.
Yes. Pardon? I believe Neha is joined.
Yes, sir.
Okay, good afternoon.
Good afternoon.
Queen Jacob has joined. Yes, good afternoon, Jacob. Good afternoon, Jacob. Am I audible, Thomas? No? Your voice is always breaking. Why? I don't know. Am I audible? Okay, fine. So, good afternoon once again.
Good afternoon.
Today, I'll be demonstrating few websites to you, and you have to also explore after me. And parallelly also you can try it out. Otherwise, you write that steps so that later you can try. Right? Am I visible clearly, or I should switch on my light?
I are visible.
Visible, okay. Because not me, that the website should be visible. Okay. So I will share few websites, few things to you. So far we have discussed about that, the computer-aided drug design protocols and their designing and development process, and few more things we need to discuss about that, how the drugs will come to the market and complete drug development process. Computationally, we have seen that the approaches which we use to design the drug computationally, in silico approach, right? But we did not discuss the complete drug development process. Are you getting? Complete drug development process. Of course, I have discussed some thing in brief in terms of that, how drugs come to the market, starting from the identifying the disease, identifying the target against the disease, identifying the compound against the disease or against the target, and then formulations, and then getting approval from IND, and then process for another agency called NDA, right? For clinical trials. Then it will go to the clinical trials first phase, second phase, third phase. Phase 1 will be that animal models, preclinical trials, and phase 2 will be the clinical trials and human models, right? Drug will be incorporated in humans, and that also population should be decided the phase wise. First phase will be that less than 100, second phase will be the less than 1000, and third phase will be the more than 1000 something. So an average first phase, the population size of the patient for the clinical trial should be 80 to 100, 80 less than 100, 80 to less than 100, then 100 to 300 second phase, and then 300 to 1000 something on the third phase. But the providing condition is that in first phase itself, a good response of the drug should be shown, right? And then second phase also good response should be shown as an output. Then only it should be introduced in that third phase. Even in second phase, first phase result should be positive. Then only can be introduced in that second phase in the big population, and then followed by the large populations, right? Okay. Then once it will give the good response in the large population, then we can file these things, getting approval from the FDA, right? Food Drug Administrations, right? Food and Drug Administrations, and then can go for that file patenting and this all, right? And then can be marketed. So these are the complete process. We'll be understanding that. But today we will be knowing that, right? Where are those information can be taken computationally? What are the resources for this? Are you getting or not? What are the resources for this? For drug type molecules. Of course, being a bioinformatician, being a computational biologist, right? And being a having a programming skill, we must maintain our databases, right? And store useful information, even the junk informations. And the expert will be retrieving those informations, right? One by one. It depends upon the requirement. So first, I will be showing you the few databases for the small molecule, where we can get the information of the drug molecule. Then we will come to that big molecules databases, right? Okay, then I will show you what are the informations are there, and those and based on those informations, what are the informations really is required for our studies, right? And you will be understanding also that what are the things we have discussed so far. So that aspects also I will be trying to make you understand this all these things, right? So I'll be sharing now. So first I will be starting for the small molecule databases. Is this visible?
Yes, sir.
Is this visible?
Yes, sir.
What is there in my page?
Drug bank.
Okay, drug
Diagonal.
bank, fine, wonderful. Okay, okay. And now?
PubChem.
Okay, fine. Now?
PDB.
Okay, fine.
Mars.
Now?
PDB sum.
Okay, fine, good. Very good. So now this is a drug bank. Already you know that, right? Okay, drug bank. Even that resources, you can say that uh, CA, that country code is there, right? Okay. So drug bank, you can say drugbank.ca. Even you can write in Google drug bank alone, you will get the page of the drug bank, right? It's clear. So now you can see here, uh, yes. Now in the drug bank, you can say that here browse option is there. In the browse, first you say that few menus are there. So in the browse, either you can run through the drugs categories, the pathways of the drug reactions, drug classification, drug target, and their pharmacogenomics, pharmacometabolomics, pharmacotranscriptomics, and pharmacoproteomics, right? Okay. So that so drug is there, their category is there, pathway is there, the reaction is there, the classification is there, and drug target is there. Pharmacogenomics, genomics is that functional genes which is responding to that particular drugs, and the metabolism of the drugs, and the transcriptomes, if any transcriptomes we are getting, with that effect of the drugs, and the proteomics, that proteins and the drugs action, reactions, functions, and all this. So new things have been added, and first time I am seeing also incorporated in this drug bank is called the COVID-19. So if you say that COVID-19, some informations will be there, of course, right? uh, so that COVID-19, all the informations are there. So what are the drugs, are how many drugs are there in the markets in the trial, not available drugs, and drug targets are there, clinical trials 1041s, and publications also 54. So even that publications are scattered in different databases, right, and different literature databases and all. But this drug bank have put their effort to bring all those papers in one domain so that researcher can see that, right? And the search option here is also is there, search for any chemical structures, search for the molecular weight, drugs, target sequence, pharmacomics, advanced search. So these all are that, and their NMR search, 2D NMR, and some wet lab experiment result is there, gas chromatographic result is there, mass spectroscopy results are there, and many others. So complete hub of the drugs and their informations are available in the database. Not only in this database, like many databases are there. I will be demonstrating few. where you can get that particular informations, right? And then if you say that now the download options, if you download many things, you can download it. For that you have to register yourself, right? All drugs, the release date, person, size, and the download entire informations I'll be downloading to this, right? And commercials data are there, clinical drug data will be there, scientific drug data will be there, industries related with the drugs, drug discovery, drugs repurposing industries will be there, precision medicines will be there, machine learning approaches and all. So all the new things has been included now, because before I'm using this tools and all last 20 years, but I have not seen these things type of machine learning and all before, right? Like within like 2-3 months it has incorporated. So it's a good indicator that all the informations pertaining to the drugs, even that through the computational methods are available for us for the reference. And help, other things are there and about, right? Help if you want anything. So here you can say that main things we are not using these, all this, most of the times we don't use, we use only these things, this, right? For getting the drug informations and all. So drugs are there, targets are there, pathways are there. Drugs, target and pathways, indications are there. So can you tell me any drugs which you know? Any drugs? Am I audible?
Yes, sir.
Yes?
Atarax,
Yeah, can you tell me any drugs which you know? Any drug name which you know?
Atarax,
Huh?
Atarax, ATARAX,
A?
ATARAX,
R-A-N. Is it correct?
ATARAX,
R-A-N-E.
RAX.
It's correct.
Okay. So,
You text me there, na? Message will be there, na?
So, yesterday's class, acetaminophen for painkiller.
Huh?
So, from yesterday's class, acetaminophen.
Okay, I'll just say that paracetamol, everybody knows, right? Paracetamol, right? Okay, everyone knows these things. It's very, or acetaminophen. You are talking about aceta. Right? So I have searched these things, acetaminophen. So now you can see that name of the drugs are there, right? Name of the drugs is there, acetaminophen, right? And here is that for this drugs, particular drugs, target in terms of that protein, 4 proteins are available where this drug is targeting. Yes, and the enzymes 15 are available, carrier as a transporters 1 is available, right? And again that here also transporters is given 1. Okay, carrier will be again the same terms, right? Will carry to the others. And bio interactions 22s are there, 22 data which are available. So now accession number, you can say that this is the unique number for the drugs, accession number, that we say that DB00316 APRD00252, and this is also called the drug card. This is the unique number to get all the informations pertaining to the acetaminophen. Are you getting? Yes. Here
yes,
we have only given the drugs. If we are looking for the target, then again we have to search for the target for acetaminophen. Pathways for the acetaminophen. Indications of the acetaminophen or particular disease. Here only we have taken for the drugs. So pertaining to the drugs, all the information will be given. Is clear? Yes. So
Yes,
this
sir.
is approved drug. Group is that approved drug, type of small molecule. Descriptions is a paracetamol. That's why I say that for any fever type of things, right? Fever. Only that paracetamol is available in the market, and the generic name will be different. It's clear.
Yes,
So,
sir.
and this is known as Tylenol, and it's a analgesic group. So you can read these things, right? Their descriptions are there because time is limited. So structure wise, if you say that their structures are also available here, you can download it, you can magnify the structure, you can download 3D structure and download it. So this form, like how I have asked you to save 3D structure in mole format. Once you download here also, they will ask you which format. Are you getting or not? And similar structures, you may find similar structures having the same scaffold. Here is one benzene ring is there. Another is that OCS3 functional groups are there, which is attached with that HN amide group, and OH is there, right? But overall, for these structures, I think benzene rings are the common. Are you getting or not? So you can find similar structures or download structures. I say that I'm downloading the structures. They'll ask you that these are the 3D structure format. Are you getting? Mole format, structure definition file. SDF is called that structure definition file. Mole is also 3D format, and it's a 3D SDF format. It's a three-dimensional structure definition file. PDB format, here itself you can download. You can download PDB format. PDB is called the protein data bank format. Smiles also you can download it. Here Smile is that one format for any structures which is attached by the parenthesis. I will show you also. INCHI is that international chemical identifier for anything is international chemical identifier, right? So these are that formats are there. I will show you all this format. So which format you are looking for? So I have asked you to mole format. Even the mole format you can download. This is the mole format. Later stage I will be demonstrating you what are the informations are there in the mole format, right? Isn't it? So it's a mole format. 3D geometries are there, three-dimensional. X, Y, and Z axis are there, and others informations I'll be showing you. This is the mole format, and what you have to do it, this format you have to download and save it. So download, and in mole format or SDF format, 3D SDF format. This is the SDF format. What you have to do it, right? What you have to do it, you just copy these things. It's a big file and put in that .sdf format, right? It's clear. .sdf format. Since you are very known for the mole format, what you have to do it, you have to download the mole format. Go to the again download options, mole format. So this is the mole format. This is the file format which you are seeing now. So these things you can copy it. Uh, uh, right click, copy it. Open your WordPad, WordPad, right? Open the WordPad, paste this structure, WordPad is visible for you, not you or not?
No, sir.
Okay. I have copied the things. Was that visible?
Yes, sir.
Yes.
Yes, sir.
Okay. So, now 1 second. Just open WordPad. Every bioinformatics things should be in WordPad. So now I will be sharing this WordPad. Now is this visible?
Yes, sir.
So in the WordPad, just you copy it from there, you paste it, right? Paste it. So now go to the file option, paste it, save as. Go to the file option, save as plain text documents or rich text documents, whatever you want, you can do it, right? Okay. So if you want plain text documents, so in that the text will come, right? So what you have to do it, okay, you save into the plain text documents. First, it will come text actually, but here you have to mention that acetaminophen, which structures you are downloading, .mol, right? So of course it will come .text. For that also options are there, mol, and where we are downloading, where we are keeping these things in our, I should say the desktop, or I say that computer aided courses, uh, value added courses, acetaminophen. Okay, right? Have you seen the process which I have done it?
Yes, sir.
Yes. Now I have saved this document here. I'll show you. I have seen documents here. If I open these things, 1 second. Is this visible for you? Which I'm doing now, opening?
No, sir.
No. Okay, 1 second. Yeah, is this visible? Is this visible?
Yes, sir.
Yeah, so acetaminophen, you can see that mole acetaminophen dot mole, right? I have saved this structure. Can you see here?
Yes, sir.
Right? Now this is the mole format, and this mole format you can visualize it, right? If I say that I should open these things, I should open with option will be there, open with more applications. Anyway, in mole format, because again I have to share these things, right? Or I have to open these things, then only we can share, like KMSketch, select, because you know the KMSketch. Right? So 3D interface. KMSketch is visible for you. Is KMSketch is visible for you?
No, sir.
No. Okay, fine. 1 second. Okay. Now is this visible?
Yes, sir.
KMSketch is visible?
Yes, sir.
Yes or no?
Yes, sir.
Yes, sir.
Okay. Now you can see that the downloaded structures which I have downloaded from the drug bank, which I kept it here. Is this visible, which I'm doing now?
Yes, sir.
Okay. So courses, I have put it in that value added courses, value added cat, and that mole format. So this is mole format. Can you see the acetaminophen?
Yes, sir.
Okay. So open. So this is the structure which we have downloaded from the drug bank.
Yes, sir.
Yes.
Yes.
Okay. Now we have justified their valency and we can save it in the same file. Save in the same file. So just I have shown you that how to download the structure even from the databases. Right? Okay, same structure I have saved it. It's clear?
Yes, sir.
Okay. So now we'll come to that. Is clear this all these things to everyone?
Yes, sir.
Yes, sir.
Okay, fine. So now come to the again for this what do you say drug bank. Again come to the drug bank now. So now we are in drug bank, right? Yes, is this visible?
Yes, sir.
Okay. Now similar structures of the drugs also you can find out, which will be having, you can say that plenty of similar structures are there. You can see here, and this is also one of the interface. Only 10 minutes are there. Okay. So here is, you can say that here also one of the domains here also mentioned. You can see here how like in ISIS draw and chem sketch also interface is there. Even you can draw the structure here, interface, right? So here three options are there for searching that similar structure. One is the similarity, another is the substructure, another is the exact structures, right? Okay. And another criteria for filtering that compounds, minimum weight and maximum weight, maximum result, how many you are expecting, and the types of the drugs which you are looking for, approved, vet approved, nutraceuticals, investigational, withdrawn drug, illicit, experimental, and things. It depends upon that you can. Now you can say that 1 to 3 results are there. So having the same type of, you see that only the changes are there in the functional group. Are you getting? Yes, these are the things you can see here. Right? 1 to 3 drugs are there, na? Yes and no?
Yes, sir.
And the 3 drugs having the same scaffold, the benzene rings. And of course, their functional groups are different. Yes? Okay? And all 3 different drugs having the different properties and having different drug cards. This is the drug cards actually. So each drug cards you can click on this. You say the acetaminophen drug card, you can go. Right? The same things you will come. Same page which we had before. So similar, the synonyms of these structures will be like this. Many structure will be there. Similar type of structures, same, and the product images are there. You can say that prescriptions, product which are available in the markets, the doses in terms of the tablet and that syrup injections and the strength, their like concentrations, right? 500 mg and this liters and this microliters or mu L or whatever nano, and route how to either is oral, intravenous and things. The labels are there for that pharmaceutical industries. Marketing when it started, marketed ended, not applicable, and few drugs. If I've shown that affect something, their market has stopped in 2012, but it still is the market. So acetaminophen tablets are there in market, but different name and brands, right? So now another is the generic definition, prescriptions are there. So there are 2 types of drugs names are there. One is the generic, another is that brand name, right? So generic name mainly it depends upon that salt which are available in that and the drugs, major constituents of the drugs, right? So those things will be there. And you can say that, and then about that plenty of informations which are available. Now we are looking for this because each information, if I told you, that will take a lot of time. You are free to explore it. I was talking about this now. Every drugs having the CS number, weight, chemical formula, INCHI, you can say that international chemical identifier. This is also one of the unique identifiers which is used globally, internationally. In India also, with this number itself, we can identify the drugs, right? Okay.
Okay, sir.
This is like scanning, which we are scanning na, on barcode. This entire descriptions can be put in the barcode, so we can scan it. So this is that international chemical identifier, right? And this is also entire international chemical identifier details. IUPAC name, already you know the IUPAC name of all the chemical compounds have the IUPAC name. And this is the SMILE notations, which we are talking about, right? And this SMILE notations also have the type, many types of notations are there, which we can understand what type of drugs are there, what type of interactions are there, and how to define different type of interactions in SMILE notation, right? And then indications are there, and the pharmacodynamics properties are there, mechanism of actions are there, how the drugs is interacting, those all these things are there. About the target informations are also there, where these drugs are targeting. These are the enzymes. You can see that synthesis, all are the enzymes. So it's a targeting. These drugs is targeting into these enzymes, and all enzymes are protein, and all enzymes and protein have their structures. We have to identify where our compound or drug type of structures are interacting to that proteins or enzymes, and those also having the structures and actions. These three for these three enzymes, these drugs work as inhibitors, and for this receptor work as an activator. As I have discussed before, like drugs will work as initiators, activators, right? Inhibitors, right? Okay.
Yes, sir.
And then the organism is human. So if say that, and then drug should have that ADMET properties, some absorption properties, their details are there, volume distribution properties should be there, protein binding. So this is are that the binding of acetaminophen to the plasma protein is low, ranging from 10 to 25 when given the therapeutic doses, right? So we know these drugs have the side effect, and the metabolism of these drugs are available. So we need to understand elimination rate, because we should not like stay long for in our body or tissues and cells. It should excrete from our bodies and tissues. So that's why the elimination rate of this acetaminophen, half-life is there, toxicity level is there. In that in toxicity, LD50, LD50 is lethal dose, right? This is that their value map 330 mg per kg oral mouse LD50 for this oral and rate. These doses are there. Overdose and liver toxicity also for this there. That's why it's a paracetamol when fevers and the body may ache or something is there. It can be like given. Otherwise, you should not take because it have the side effect. It's a carcinogenesis, mutagenesis properties are there, right? So we have to take care about these drugs. So all drugs, and now you can say that control F, you find, and many literatures are there about these drugs. I don't think only 2 minutes are there. I don't think we will be able to complete even the one website. Then ATC codes are there. ATC codes, we say that anatomical therapeutic chemicals. Actually, what the major chemicals which are available in the drugs, which are there here, right? So this is the ATC. Drug can be categorized based on the ATC also, and of course other parameters are also there. But ATC is that anatomical therapeutic chemicals. Anatomical therapeutic chemicals. So in that category, we can dividing our drugs and the PDB entries. So these are the PDB entries are there. Even show more. So in this PDB entries, these are the PDB structure actually, protein structure. I will be showing in that now, protein data bank. These are the protein structures where these drugs having the interactions, right? And that interaction I'll be showing only 1 minute there. I'm not able to show you now. Maybe tomorrow I'll be showing you. You write it this at least the PDB entries. 1 TYL, tomorrow I'll ask you. 1 and 2 you write it, please. Am I audible?
Yes, sir.
Yeah, you write it 1 TYL, 1 TYM, 2D PZ. I will be showing you these things. They are interactions which are available in the protein data bank. And like that, even that, more things you can search it. And then clinical trial informations are there in all the phases. The pharmacogenomics, pharmacoeconomics features are there related to the pharmaceutical industries. And the dosage informations are there. And the price. And if you say that this is the big files, so many more informations are there. So of course, time is not permitting me to all these things. If you have any questions, and even you can explore it, you can see after this, you can explore this website and see that what type of information